My name is Ikenna Kingsley Uchendu. I'm a lecturer with the Department of Medical Laboratory Sciences, University of Nigeria. The title of my presentation is Anti-Dyslepidemic and Cardioprotective Effects of Turmeric in Rats Fed with a High Cholesterol Diet. Nutrition interacts with organs in the body in many ways, and the possible effect of slowing down organ damage has been considered previously. Foods with therapeutic potentials provide important sources of most of the world's pharmaceutical. Turmeric, also known as Curcuma longa, belongs to the ginger family of herbs known as Zingiberaceae. Its use in folklore medicine to treat numerous diseases is well documented. This is the image of turmeric. The aim of this study was to evaluate the anti-dyslipidemic and cardioprotective effects of aqueous extract of Curcuma longa in rats fed with a high cholesterol diet. Methodology. Fresh samples of turmeric were purchased from a local market in Enugu, Nigeria. The preparation of the aqueous extract was done as described by Ate et al. Procedures outlined by Trees and Evans were employed for the phytochemical analysis of the extract. The experimental protocol for this study was approved by the Institutional Ethics Committee at the Department of Animal Science, University of Nigeria, Nsukka. Experimental design. A total of 20 rats were used for the study, and they were grouped into 4 groups: Group A, B, C, and D. Group A served as the negative control or the toxic control group, and the group received 2000 mg per kilogram oral dosage of high cholesterol diets, which comprise cholesterol and bile salts, and 60 mg per kilogram oral of carbimazole. Carbimazole is an anti-thyroid drug, and it is known to induce hypercholesterolemia. And its use in this research was to enhance buildup of cholesterol in the blood. Now, Group B, which served as the positive control, they received the standard lipid lowering drug known as atorvastatin, 20 mg per kilogram oral, along with the toxic substances. And Group C, which served as the test group or the experimental group, received 400 mg per kilogram oral dosage of the extract along with the toxic substance. Now, Group D served as the normal control, and this group received no treatment throughout the experimental period. And the experiment lasted for 8 weeks. After 8 weeks, the animals were sacrificed under anesthesia. Biochemical markers of lipid profile, which are total cholesterol, triglycerides, and high-density lipoprotein, were assayed using standard methods. The hearts were harvested for histopathological analysis. This is the result of a preliminary phytochemical analysis of the extract. The result shows that flavonoids, tannins, phenols, terpenoids, and phenolic compounds are present in moderate amounts, while saponin and steroids were present in trace amounts. And we had total absence of glycosides in the extract. This is the result of the Lipoprophyte analysis. From the result, it can be seen that the negative control group showed significant elevation in the total cholesterol and triglyceride, and significant decrease in the high density lipoprotein when compared to the normal control group. This is the photomicrograph of the heart section among the groups. From the result, it shows that group A, which is the negative control group, has significant damage in the cardiac fibers, showing significant fibrosis and inflammatory cells. Red arrow. Now the group that received the standard lipid lowering drug, group B, showed significant protection in the cardiac fiber, as can be seen here, with mild infiltration of inflammatory cells. Now group C, which is the group that received the aqueous extract of the Curcuma longa, there is protection in the fibers, but not as was seen in the positive control group. Now group D was the normal control group. The cardiac fibers appeared normal. Discussion. The effect observed in this study could be due to the singular or combined action of the bioactive phytochemicals present in turmeric extract. Hyperlipidemia and oxidative stress following hypercholesterolemic diet could be prevented by endogenous and exogenous antioxidants. The consumption of plant products was shown to reduce hypercholesterolemia, oxidative stress, homocystinemia, endothelial dysfunction, and blood pressure in previous studies. These beneficial effects have been partly attributed to the compound which possesses antioxidation. Turmeric is rich in both anti-inflammatory and antioxidant phytochemicals such as flavonoids. Conclusion. This study showed that the aqueous extract of Curcuma longa has effect on lipid metabolism and prevents cardiomyopathy in albino rats fed with a high cholesterol diet and high dose carbimazole. Therefore, the alterations in the serum liquid by the aqueous extract of Curcuma longa may not predispose the heart to atherosclerosis or its associated cardiomyopathy or infarction. Thank you for listening.